All right, so today we're going to be talking about biotechnology and how we use the information we've talked about in our genetics unit to actually apply it to the real world and do some of the creative things that we do in science and medicine. I feel like this is a very interesting topic just because it's kind of like the ground line of a lot of work that's being done in science these days. So biotechnology is simply the genetic manipulation of organisms, and humans have been doing this for thousands of years. Today, we actually have the technology to go in and actually edit the genes of organisms. But in the past, farmers have been doing it by selective breeding for plants and animal species. For instance, farmers will select two types of plants to breed together in the hopes of having a food source that's going to be larger and have more actual sustenance for humans to consume. The same thing with farm animals. They'll breed two very large cows together, knowing the offspring is going to be large, therefore, it will be a great food source when the cows go out for slaughter. Um, so it's kind of interesting when you think about going to the grocery store and buying even just produce. If we were to Google an image of a natural banana versus one that's been genetically modified, you'd kind of be shocked. Here's a good image comparison here. So here's a normal banana like you'd think you'd buy at the grocery store. But if you look at a natural banana, here is a natural banana, and a natural banana has less actual food and a lot more seeds. It's much smaller, has much larger seeds, and this wouldn't be something you'd want to eat. You wouldn't actually get much food out of it. But it's been modified such that now it's much larger. The seeds are really small. You wouldn't even really notice a banana has seeds, but there are. They're really small right there in the middle. And it's much better to eat. And that's true of a lot of different produce that we have these days. They've been bred, selectively bred, such that it gives humans more food consumption. And the current or future use of a lot of this genetic technology is to make medicines, vaccines, cloning, modifying organisms to give them genes that we want them to have that they may not already have. And here's a picture of a glow in the dark mouse. That's actually been used to do like brain mapping using glow proteins to actually trace where things happen in the body. So it's a quite interesting topic to discuss. So we're going to start with just the basic toolkit of some of the things we're going to talk about. So basic toolkit that we're going to talk about. The first thing is restriction enzymes. Restriction enzymes are kind of like little DNA scissors. One example of a restriction enzyme and how it's going to be used is with another tool called ligase. We talked about ligase way back when we did DNA replication. Ligase is a enzyme that glues things back together, glues DNA back together. We're also going to talk about plasmids and cloning. In our advanced DNA technology toolkit, we're going to talk about PCR. PCR makes copies of DNA. DNA sequencing to get a specific DNA profile for an individual. We're also going to talk about gel electrophoresis. Gel electrophoresis is going to allow us to make DNA profiles to figure out who left DNA at a crime scene or possible paternity. And we're not really going to talk about this last one, but it's another thing that's often used. It's called a southern blot, and that is what type of blood test. So genetic engineering is the process of manipulating genes and DNA for a practical purpose, those that humans deem are practical or beneficial for our well-being. And genetic manipulation may involve recombinant DNA technology, where we actually recombine DNA from different organisms and make it into DNA that we want an organism to have. So we take a gene from one organism and put it in another to make an organism that can do something that we deem would be beneficial for them to do. And we're going to talk about exactly how that's done. But just a little bit about the terminology behind taking a gene from one organism and putting it into another is recombinant DNA is the actual DNA itself that's been recombined. So recombinant DNA is the actual DNA that's been pieced together from one organism or another and put together to make the DNA that we wish. Whereas an actual organism that contains the recombinant DNA is known as transgenic. It's transgenic because we transferred a gene into it. So we transferred a gene into it, so it's a transgenic organism, but the actual DNA itself is recombinant DNA because we recombined it to make it unique. Now, general steps that we're going to use in this process. The first step that we're going to do is cut out the gene that we want. Then we are going to make recombinant DNA. Then we're going to clone it, make lots of copies of it, and then we're going to screen it to help purify it. So we're going to talk through these one at a time. So the first step is cutting the DNA, and cutting the DNA is done by restriction enzymes. So restriction enzymes are like little DNA scissors. Their job is to cut the DNA at a specific place. So restriction enzymes cut DNA at a specific place. And if you look at these sentences we have here, these are palindromes, meaning they're the same forward as they are backwards. Very similarly, in DNA, the restriction enzyme is going to look for a particular palindrome and cut there. So if here's our DNA over here to the side, highlighted in red is the sequence that the restriction enzyme is looking for. Along the top, if you read it forwards, it's GAATTC. Along the bottom, if you read it backwards, it's GAATTC. So it's the same forwards as it is backwards on the bottom. So the restriction enzyme will look for that specific site, and it's called the restriction site. And that's where it's going to actually cut. The restriction site is the place that the restriction enzyme looks for, and it's where it cuts. And when it cuts, it's going to cut at a weird angle. It's going to cut. So if here's our sequence, let me work on writing here. So if we're looking for GAATTC along the bottom, it's going to be similar. Just like this. So once it finds the sequence, it's going to cut between the G and the A. So it cuts between G and A on the top and the bottom. But it doesn't cut all the way across, just between the G and the A. And it's going to cut at an angle like this. So our pieces of DNA in the end, one piece is going to look like this, and the other piece is going to look like this. And it creates what is known as sticky ends. And it's very important that these sticky ends exist because when DNA is recombined, so when we actually combine the DNA and put that DNA back where we want it, DNA doesn't attach side by side like this. It attaches top to bottom like this because A's and T's go with G's and C's. They don't stick together side by side, they stick together top and bottom. So it's very important that we have those overarching overhangs so that the DNA can base pair back together in the end. And then, after we have our sticky ends, we're going to mix that gene once we cut it out into our plasmid where we want it, and then it's going to combine back together where we want it. So we cut out our gene, we leave a sticky end, so a little overhang like we see here. Now our DNA is cut out. Now we have to paste it where we want it. And for this, we're going to need what is called a vector. Now, a vector is some type of special agent that's going to carry the gene that we want and put it into another cell. So the vectors that we are going to talk about in this class are plasmids. Plasmids are circular bacterial DNA, and they can carry the gene that we want into the organism we want it in. Now, plasmids, they're very, very helpful for a few reasons. One, they are small, circular pieces of DNA. Again, initially they are found in bacteria. And they are self-replicating, meaning they make copies all on their own. So that's going to take a lot of the work out for us as scientists, because if the DNA in the bacteria is going to copy all on its own, we don't have to help with that job. It's going to go ahead and copy itself. It also, the plasmid, has a gene for antibiotic resistance, meaning antibiotics won't be able to kill it. And if antibiotics can't kill it, then we know that our gene will be safe. And that's going to be very important here a little later. And also, plasmids, we can easily paste the information we want into them. So what we are going to do, we're going to use our restriction enzyme to cut out our gene. That was step 1. Step 2 is going to be to put that gene into our plasmid or vector. So we're going to use the same restriction enzyme to cut a hole in our vector. And then we're going to make recombinant DNA. So the way this works is if I have my vector here and I have my gene of interest all cut out here, I cut them with the same restriction enzyme. So this one's been cut out. Whoa, that was not the one I wanted. So there we go. We cut here with the restriction enzyme. We've also cut this open with the restriction enzyme. I'm going to mix the two together. So when I mix the two together, what's going to happen is my gene that I want is going to attach right into that vector. And now I have my recombinant DNA. So I cut out my gene. I cut out a little spot in the vector. I mix them together. And now I have the recombinant DNA. Once I mix them together, I need to be sure that we use ligase as a glue to glue that gene in place. So now it's in my vector and I have my recombinant DNA. Now that I have my recombinant DNA, I need to make copies of it. So now that we've made the gene that we want, we've put it in the right place, we have to make copies of it. Now, it's very important here. We said that the bacteria are going to make the copies for us, so we just let them do their job. Nice and happy. We don't have to do much of anything. Step 4 is screening. Screening can also be thought of as purification. Because if we're making a vaccine or something, we want to be sure that it's pure and it's not contaminated with any extra bits and pieces. Because when we cut out our gene and we cut our vector, sometimes there might be some extra scrap parts that don't fit together, and we need to be sure that we sort all those out. We need to purify our gene of interest that we put in our plasmid. And the way that we do that is by taking the bacteria and putting them on a plate with antibiotics. The antibiotics are going to kill everything except for the bacteria that has the gene. The reason that is because the one with our gene is antibiotic resistant. So we know that anything that grows on that plate is going to be holding the gene that we want. Now, some uses for things like this. Here's our little image here. We'll go through that again. Here's our plasmid. We cut a hole in the plasmid. We cut out the gene that we want. We mix them together to make recombinant DNA. Bacteria will copy it for us. We purify it by growing it on an antibiotic plate. And then we have our whatever vaccine it is that we are creating. And again, this is used to create genetically modified organisms, also known as GMOs. It's used to create a lot of the plants that we have and foods that we buy. It's also used in the healthcare field to create drugs, vaccines, and gene therapies. And it's also used to do cloning, which has been done on farm animals as well as pets. I know there was a few celebrities that cloned their dog and got really disappointed when the dog didn't have the same behavior as the one before it. And what they didn't account for is that a lot of behavior is created by what you're exposed to, your environment. So the dog isn't raised identically to the one before it, they're not going to turn out the same. So that was creating recombinant DNA, and that process would be used to make, say, a glow-in-the-dark mouse. We would take the glow gene from a jellyfish, do this process to create a glow-in-the-dark mouse. This could be used to create a vaccine. So if we wanted to create an insulin vaccine, we could take the insulin gene out of one organism and implant it into another. So this can be very useful for a lot of different things. The last thing that we're going to mention here in biotechnology is about using DNA gel electrophoresis. So, fun fact, it uses gel and it also uses electricity, gel electrophoresis. What gel electrophoresis is going to do is it's going to take DNA, chop it all up into pieces, and then sort DNA out by size. And this is going to be incredibly useful. So the way that it is done is we use DNA to cut we use restriction enzymes to cut the DNA into various pieces. Once the DNA is all cut up into various pieces, we load it into a gel. It's just like a little jelly plate, probably smaller than your phone, a little smaller than your phone. And we inject the DNA into the little wells at the top. So here's an image of the DNA being injected into the wells inside of a gel. Once the DNA is inside, an electric current is added. And the reason that this works is because DNA is negative in charge. And since DNA is negative in charge, if we inject DNA right here on the negative side, and then we apply electricity, DNA is going to move from negative end to the positive end, and it's going to separate. And the reason that it separates is because large pieces of DNA are too big to move quickly. Big things move slowly. So the large pieces are stuck at the top end, and then the smaller pieces are able to move farther in the same amount of time, so they migrate down. So it separates the DNA out by size. And when you do that, it creates a unique DNA profile. So every person is going to have their unique DNA profile when gel electrophoresis is done. And this will help determine several things, the first of which is if they find DNA at a crime scene, they can take the crime scene DNA, make sure it's not the victim's. Here's the victim for comparison. And then they can see which suspect left their DNA at the crime scene. Now, because this is one person leaving their DNA, they either leave all of their DNA or they leave none of it. So it's going to be a perfect match. So in this case, the perfect match is suspect number 2. So suspect number 2 left their DNA at the crime scene. That would mean suspect number 2 would be considered guilty. This can also be used, and I don't have an example here in this PowerPoint, but it can also be used to determine paternity. So which one is more likely the father. And the way they do that would be they would have a line for the mother, a line for the child, and then the potential fathers. Now, there's a little bit of difference between doing crime scene and doing paternity. At a crime scene, they match 100%. In a paternity test, they're only going to match 50%. So, for instance, if we were looking at a profile, let's just use the one we have here, and say this line here was for the child. This line either came from the mother or the father. So each line comes from either the mother or the father. If this line does not match with the mother, then it has to match with the father. So the way that you would figure out the father of a child based on an gel electrophoresis is you would look at the child and pick out which lines that it had that the mom didn't. And you only look at those because if mom doesn't have them, they had to come from dad. And then you match those lines with dad. So it's not going to be 100%, it'll be roughly 50%. Okay, so there's going to be some questions about this on Canvas, so go ahead and